So hello everybody, welcome to our journal club. Thiago helped me to pick this manuscript, this paper to present to you guys. Well, first of all, it provides us data on the single cell RNA-seq, and they also provide us data with immunohistochemistry proteomics. Okay, and the first this was published last year, and it presents to us the protein expression profile of angiotensin conversion enzyme 2 in human tissues, how this protein is distributed in our body. Okay, and why this is important, because they wanted to understand the host susceptibility to SARS-CoV-2 infection. Well, to understand this, we need to know where these receptors are. Why? Well, they decided to study these because this is the virus, right? The SARS-CoV-2 or SARS-CoV virus were shown to enter the cell through these receptors, okay? And they are primed by cellular serine protease TMPRSS2, okay? And reading the abstract, you will notice that the ACE2 expression was observed in enterocytes, renal tubes, gallbladder, cardiomyocytes, etc., etc., etc. But however, in the respiratory system, the expression was really, really limited, with no or only low expression in a subset of cells in a few individuals. This is very curious observation. When I read this, I was like, oh, come on, this is a respiratory virus. How come? It goes in, like it enters the human body through the respiratory system, and it's been demonstrated before. This one here, this Hoffman paper, is a very interesting paper on how the molecular mechanisms of how the virus enters the cell, right? I didn't read this one, but I was really, really curious about how come this is possible. I mean, if the virus enters through the respiratory systems, through this receptor, but the receptor is in low expression in cells of the respiratory systems, how come this is happening? And I was really curious. That's why I decided to read fully this paper. So the results they are presenting here are basically the expression level of ACE2 at the transcriptomic level, at the single cell level, okay, and protein profiling based on immunohistochemistry. I was searching actually for image analysis of COVID-19 infections, and I found this paper. But reading the abstract was really, really what made me go deeper on it. So for the gene expression profile, they gather data from three different databases: the Human Protein Atlas, this one, I'm sorry, I forgot the name, and the function annotation of the mammalian genome. Okay, all the data is available, and they have gene expression from different tissues of the human body. And here they present the first results they are presenting is this normalized expression index that they gathered between all these three databases. So what they found is that the higher expression levels are in small intestines. Yes, some questions. Someone drop a question. Genotype. Okay, thank you, Amanda. Genotype tissue expressions. I forgot the name, but yeah. So they found that the most tissues that express this is really in the small intestine. And lower expression, like in other tissues, but kidney also, and testis, and gallbladder, heart, so on and on. Okay, so this is the expression based on tissue transcriptomics. Now, for the single cell part, this is a really long figure. It's almost in the whole. I hope you guys have the paper, looking at the paper at the same time as me. But then I just highlighted a few parts of the text. But again, the single cell, basically it confirms like the results were largely consistent with the transcriptomics from the other data sets, right? So showing higher expression in ileoenterocytes, of course, so cells from the gut, and renal proximal tubes. So here are the findings, and enterocytes, like higher expression. And again, they provide from healthy individuals. That's another thing that I found interesting in this paper. Like, so, and the other, okay, oh, and then they provide another, they go deeper on respiratory tissues. Like, okay, let's see the proportion of cells that are increasing the expression, have increased the expression. And alveolar cells in the lung were the most, but here have a higher proportion of ACE2 expression. But here, if when we look at it, we see that they are using only 1% of the cells. So it's really a small percentage of cells that present higher expression of ACE2 receptors for the virus. So, and going further now, and here they is a summary of the cells that present higher expression at protein level of the ACE2 receptors, and cardiomyocytes, proximal tubules, gallbladder, enterocytes, endothelial cells. This is important. Ciliated cells, like in the nose or in the throat. And in the end, they come up with these normalized expression levels at the of proteins from different cells, from different tissues. So cardiomyocytes presented higher expression, enterocytes in the duodenum, and so on and on. Oh, also enterocytes, kidney. Well, this is quite interesting, but still doesn't answer that question that I had in my mind before. Not sure if you guys have had the same question. Okay, so this is based on immunohistochemistry. Like, so they decided to go a little deeper, using more samples from individuals to the immunohistochemical part analysis of ACE2. And what is interesting is that the results from bronchus and nasal mucosa were consistent with initial the histochemistry analysis, like tissue microarray, with cilia staining for only one of the antibodies. They tested two different antibodies in this paper, right? For a small subset of cells in few individuals. So this is really complicated, right? So still, how come this is happening? And they also tested proteomics databases and Western blot. Like, this is the last result they present. But looking at the histochemistry, right? What we see is like, okay, we see like in the enterocytes, we see very high expression, testing two different antibodies because they validate antibodies. Part of this initiative, the Human Protein Atlas, what they do, they validate antibodies. I'm not sure if I'm being clear with this. If you guys have any questions about how this happens, they provide a small description in the discussion of the paper about the importance of validating antibodies to get to make sure that you are labeling the right protein in your sample. So in some tissues, they didn't find an agreement. For instance, down here in the epithelium of the fallopian tubes, there is no agreement between the different antibodies they test. Another, in their rectum, they also provide some positivity, right? You can see that the cell is positive because there is this brownish label here. Not sure how many of you guys are familiar with immunohistochemistry, but this is a really interesting images to analyze. And so this is quite very intriguing. What kind of cell types, but they are labeled. But for kidney, well, there's a high expression of ACE2, and in the heart, they also didn't find any association between the two antibodies. There's a difference in the expression, but in general, it's a high expression. Now, specifically looking at tissues from respiratory system, the respiratory tract, the epithelial mucosa, like nasal mucosa, the epithelial cells are with higher expression. The ciliated cells are the most, basal ciliated cells are the most with higher expression, as we can see pointed in these arrows here. And some rare cells are present here. This is, sorry, I forgot this. Cell type localization specific with based on human histochemistry. So, yeah, AC2 could be observed at the base of ciliated cells, both in ciliated cells. Rare AC2 staining was present in a few alveolar cells. So these cells here, right? That's quite interesting, but still doesn't answer the question. And well, it shows us how important this kind of study is, because we are mapping the human body of proteins. So these are all healthy individuals. The quantification of this is in this table, showing how rare it is for the expression in lung tissue microarrays. Okay, and so on and on. And then they begin to talk about, in the discussion of the paper, how they validate the antibody. Okay, they tested two different antibodies to generate the images and quantify the expression patterns. The big advantage of using immunohistochemistry is because you can have a semi-quantitative expression and also the location of the expression. That's the biggest advantage. And it's interesting when you use this kind of technique, because you confirm the findings of RNA-seq, single-cell RNA-seq that we have, such as this one here in the second figure of the paper. So it was really, really paper I really enjoyed reading it, and I learned something here. And then after they providing like the overall expression protein patterns showed a high correlation with mRNA expression from the different data sets. They also highlight that the tissue where the proteins are found, like it is summarized in here, right? These tissues are also tissues where patients with where the virus, where patients are reported with highest damage. I mean, sorry, English words are fading from my mind, but let me say quickly in Portuguese. O que eles associaram aqui foram que os tecidos onde encontraram maior quantidade de proteína, ACE2, são justamente os tecidos onde estão mais associados os sintomas com SARS-CoV-2. Então, assim, patients with worst prognosis presents, several recent studies have highlighted COVID-19 related symptoms in organs expressing high levels of ACA2, including gastrointestinal symptoms, renal failure, cardiac injury, and effects on male gonadal function. But of course, this paper was published on June last year, and many of these papers that about the symptoms they are citing here are preprint. We should check if they have already been published or revised, but of course, we are talking about clinical findings associated that with the presence of the tissue. So this is really important argument in favor of this kind of exploration. Several organs showed low expression. Well, of course, and further colocalization studies using high-resolution microscopy are needed in order to confirm the vascular expression identified here is in fact endothelial cells pericytes. They hypothesize that the general inflammation caused by the virus comes from infection, like invasion of the parasites. Parasites are those little cells that are attached to the vasculature, right? So they provide higher, if these kind of cells have higher expression of ACA2 proteins, so they probably have it's a target for the virus, and they are the ones responsible for generalized inflammation caused by the virus. Oh, here's the big figure about the transcripts per million. This is the proteomic analysis. This is also proteomics, and the Western blot, what they highlight is that no band was observed for the lungs, which is okay, since the virus, since COVID-19 is a disease that affects the lungs, why there is none of this protein? They affect mostly ciliated cells, and that's why we can see. So that's why we can see the fact that swabs from throat and nose, which are highly populated by ciliated cells, often contain high levels of SARS-CoV-2. And to finish, now that's where come the answer for my question at the beginning was, well, the viral, there was something that they argue here, that in a recent study that this Human Cell Atlas Lung Biological Network and co-workers established that an interferon stimulation in fact leads to the overexpression in bronchial cells, among other cells. This is this single part of the text explains everything in my point of view. Because if you think about that any viral infection increases the interferon expression, increases the interferon release in the bloodstream, for instance, microvasculature, and interferon leads to overexpression of ACA2 in bronchial cells or other kind of lung cells. So the same mechanisms that it's the same mechanism that our body uses to fight the virus infection, which is the production of interferon, also leads to the overexpression of ACA2, which as a consequence leads to the more susceptibility of viral infection. This was really blow my mind when I read this. Yeah. So receptors is an interferon gene You see, SARS-CoV-2 receptor ACE2 is an interferon-stimulated gene in human airway epithelial cells and is detected in specific cells subsets across tissues. So this is another interesting paper to read later. In the end, I think this was the answer of my. And of course, further studies are needed to investigate the limited staining or other kinds of antibodies, or to which kind of data represents the true expression. How is this upregulated, which is very interesting. All the data is available of the study and the pipelines they use. This is really cool website because they have patterns of expression of proteins from different cells, different tissues, and you can get the images. You can analyze the images yourself. You can get those images in high resolution. You can pick by organ or systems or cell type. It's a very interesting database, especially if you are really into tissue image analysis, like they provide in using tissue microarray, including survival and on other stuff. I guess that's all I wanted to say about this. Oh yeah, I did write some. One of the questions I'm asking if there is a correlation between these proteases and ACE2, because one of the reasons is that the virus doesn't go inside the cell by itself. It also needs it needs another proteases, like the spike viral protein needs these other proteases in order to enter the cell, the host cell. So, and not only this one, some other proteins were also, they also say here in this manuscript, it's right in the introduction. Yeah, here. They also say that they employ other proteases for host cell entry, including catepsin, which are enzymes of the lysosomes, and TMPRSS2. So that's why I'm asking if these catepsins or these other proteases are related. I was just looking at this before. That serine protease. It is a transmembrane serine protease. So if these are also correlated with ACE2 expression, right? This is one of the questions. Is there a correlation between these proteases and the patient outcome, like clinical susceptibility of the virus? Do you guys who are looking at RNA-seq data sets, do you know if these other proteases show up when you analyze them? If there's a differential expression, if they appear there, because, and of course, what is the distribution in the body of these proteases? That's kind of questions was really intriguing, and I was like, okay, how can we go further?
Mauro,
Yeah.
can you use the human protein atlas to show these genes distribution in the body? Just to show us how to use the website and what possibilities we have with the human protein atlas, please.
So the Human Protein Atlas is a database with images of cells, because they are mapping the location of the proteins in the human body. So we have images of tissues, we have images of cells, individual cells, and we have this is new for me. I haven't seen this before. Metabolic atlas. And a pathology atlas, like the difference in tissue, in healthy tissue and a disease, like a sick tissue, okay? It's basically searching for your protein of interest, for, catepsin B. If we give a search here, they will bring us the data. So you can download the data as many other databases, right? You have data files here. They probably have an API where you can systematically access these databases. I haven't explored that part, but let's see, cathepsin B, what other diseases this protein is associated? They have also validated antibodies if you want to do some tissue analysis. Like here is the distribution based on, oh, this is cancer. The distribution of, wow, high expression in thyroid cancer. And they have the protein expression, so the RNA expression profile, prognostic in case of cancer. So, yeah, they show me cancer because I hit on pathology. So, and images of the patterns of expression. They have 4 sets here: colorectal, breast, prostate, and lung, just to show how hard it is to get samples from other tissues. So it's not always available. When it is, they present it here. And they also presented expression summary of this. Let's go let's take a look at the cell level, how they are. All the data is curated, okay? So they have this cell scheme, which I think is very cool. And you can download these schemes and use in your presentation if you're interested, if you have a gene of interesting or a protein that you're a fan of. And showing in which organelles, which parts of the cells these proteins are detected, right? And you can validate with looking at images where there's a staining, and this is really, really interesting. So basically, that's how I use it. I was before I was using mostly to search for the images of tissues and samples. I was studying, my protein of interest was whoops, that's not a, I was studying this associated protein in cancer cells, which is a very low expression profile in cancer. However, it has a high expression at the protein level. This is the kind of protein that, so this database, I wasn't aware of this before, but this database is really, really nice. I hope you guys like it to explore a little bit, get the differential genes that you found, and just take a look here, right? In the blood, how is it going to be expressed? Yeah, well, in the blood you don't have location because it's not a solid, but it's spread in basophils. We should go see if it's monocytes. There's a high expression of cathepsin B in monocytes. What an yeah. Before classical monocytes, non-classical monocytes. Then you have to check what do they mean by classical monocytes, and yeah, how this data is curated. Detection in plasma based on, we don't have image with plasma, but
if you put the last gene, the CTSL gene, in the Human Protein Atlas, you will see that this protein is highly expressed in lung macrophages. So it's very interesting as well. Which was the other gene that the virus uses to get into the cells. So it has like a few genes that uses to get into different kinds of cells, depending on the cells that are present at the site. So it's very interesting. So this kind of information we wouldn't have so easily if we didn't have this website, right?
Yeah. They even have a game, like, because part of image analysis requires labeling, right? Requires that some, for instance, a pathologist or someone trained in reading this kind of images must look at the image and validate, well, yeah, this is a cell and this is spread in the cytoplasm, not in the nucleus, so on and on. So this requires labeling. This kind of every image analysis, Thiago, you know very well how it requires labeling. And it's a crucial step for image analysis. The point is that they created a game to train people how to label those cells, how to label those images. I just can't. I was trying to remember what was the name of the game. Like, the game is really, really cool. It's simple game that you open an image in the website, and then you just go clicking, and they present some options where you can see if that's a protein, your protein of interest or not. At the same time you are playing the game, that you are labeling the cells for the images. They don't have labels yet, and you go scoring and stuff like that, and people have like competitions of this game. Project Discovery is the name of the game. Yeah, this is basically how to install and play the game, or it's online.
This one?
Let me see. Yeah, here's an example of how the game is playing with. In a sense, you are kind of like being like a mechanical turkey, annotating images of cells or proteins in. But yeah, you can try. So there's also a high expression in monocytes, huh? They use this score, normalized expression score, because since they are gathering data from different databases, they have to get to a consensus, of course. Yeah, resulting in normalization pipeline. So this is basically how they normalize the transcriptomics data using these data sets. And that's it. You guys have more questions?
Here.
Yeah, so what do you guys think about the paper? Anybody have any comments? Patricia, you can use this website to look for genes that you found on your thesis.
Yeah,
That you
I
can
was
there.
looking here these genes.
I was looking at the SRY, which is a sex determinant region Y gene that Amanda was discussing yesterday with me. So you can find a lot of interesting information of genes that you find in your studies.
That's true.
Yeah, Leandro.
Hello, how are
Hi,
you?
Leandro.
Hi.
Hi.
There's
Everyone
also
is okay? Okay. First, I would like to say nice talk. Some very good paper. So you did well to take this paper to show us. The question is, what is your opinion? Why? What is going to be the main reason? Because there is RNA expression, but there is not an antibody. You know that you remember that you show part of the data where they compare expression with the antibody, and they do not overlap. So what could be the reason for that? The quality of data, because if you have the expression, why there is not a reason? Because of there is not a antibody.
Kind of agree. There is an agreement between the expression levels and the protein level, the mRNA level. They presented this. What is intriguing is that there is a low expression in lung tissues or respiratory tract, except by one type of cell, the ciliated cells in the epithelium. Finding is very interesting because there is this huge amount of cells, but only one is responsible. At least from this data, we can infer that is only one, a small proportion. And even so, it is in a small proportion. So I'm not sure if I'm following your question, because they kind of agree. The expression levels
No,
between.
because I saw, yes, I saw some result where protein levels is not the same with RNA expression, but you said that is a great agreement because it probably the reason because sometimes there is not expression of or there is not a great agreement between these two expression in terms of protein in RNA is because sometimes I believe that ACA2 can express in a small proportion of cells.
Mhm.
So time because this paper show us that the distribution is not depend of the tissue, it's depend of the cell type,
Yes.
and that is quite interesting. This is this is probably the expression is higher in a small intestine, but when you look in the cell, what kind of cell had this expression? Only a small proportion has this ACA2. So it's like it's quite interesting, and probably this is gonna be the reason because there is a small proportion or a small expression in lung. Probably this could be the reason. Or you have another hypothesis?
I will stick to the hypothesis that the virus is inducing an increase in the expression. I will go just like in the discussion of the paper. Virus is inducing the overexpression of ACE2 receptors um, based on the interferon response. That's I haven't read. I did not read the reference they showed. It's a Cell paper, one of those huge Cell papers. But I would go in that way. That's the most likely explanation, right? The virus is inducing the expression of ACA2, so it can go inside of the cell. That's really interesting. Let's see, Edson, I understood that in general, comparing all organs between them and AC2 do not express too much in the lung, but usually when we compare the disease, that's a good question. I don't know. I was going to ask this to you guys who are comparing. Edson, are you there? Yes, he's here. So I was going to ask you guys this because you guys are comparing and healthy and sick people. And do you see if there is an increase, a differential expression between
Yes.
those groups?
Mhm.
Uh,
Usually, when we compare the disease group against the control group, we can see this upregulation independent of the tissue. So, this is the reason why I don't think that when you see the overview of the expression in the body, okay,
uh-huh.
the lung do not express too much, the AC2, but when you compare the person that is in disease group against the health person, this gene is expressed much more in the disease group. But I made this analysis in the lungs and in intestine, I think, in your project, Leandro. I forget the tissue.
Mm,
Esophagus. Remember esophagus that we did analysis in this? Can you hear me?
Yeah,
yeah.
Hello?
Hello.
Yes.
it's esophagus.
So,
Esophagus.
yes, esophagus.
Yes. Mm,
But anyway, it's just a point.
yeah, yeah. This is really really interesting. ISG, ISG. Yeah, serine protease. Yeah, ACE1 is a protease. It helps to regulate parts of in the regulation of the blood pressure, right?
Because I never heard before that ACE2 was ISG, stimulating interference stimulated genes. This is the first time that I heard about it. So this is the reason why that I am a little surprised, but it's okay.
Oh, well, I didn't
Because
know that either. That's let me paste here the two. Where is this? Is a structure? Okay.
Esse artigo que sugere o ACE2 como um ISG, ele já foi citado 936 vezes.
So this one is the viral, and the other one is, yeah, interferon.
Vivi, você que já analisou vários datasets relacionados à resposta imune e sabe bem essas coisas de ACG, você já tinha notado esse ACE2 aparecendo nos seus dados?
Eu não cheguei a analisar o dataset, eu só estou analisando dados de literatura, né? Dataset eu não cheguei a... Mas no trabalho da Amanda e do Juan, vocês observaram alguma coisa em relação a isso, não observaram?
Dos ESGs?
Não, do ACE2, se o ACE2 aparecer.
Como upregulado nos pacientes?
A gente não olhou a nível de ACE2 porque o dataset original de onde a gente pegou os dados fez um tipo de sequenciamento muito restrito. Eu vou até conferir agora no website que a gente criou, se está aparecendo lá.
Que, por exemplo, no meu doutorado eu estudei, in my doctorate I studied the infection of fibroblasts during the infection with T. cruz, and I noted a bunch of IS genes upregulated, but I didn't note the AT2. So anyway. Maybe is a point for we check, we guys that are working with transcriptome data, check, and when we see these interference stimulated genes appearing like as an enrichment, functional enrichment, we can check if ACE2 is also there, because maybe this gene was ours there and we didn't note it.
Bem, gostaram?
Muito.
Sim, muito obrigado.
Sim, muito demais.